Morning. Hope you all enjoyed the weekend. So today we're going to cover really the last bits of new content. And I've labeled this lecture Diffuse Projections, even though I'm going to actually start by talking about something that really doesn't fall into that category, just because I've dropped it twice at the tail end of two other talks. This is still not working. I still have to fix that. All right. What we've already covered now is really everything that shows up on the slide. So we've dealt with how you do all of that receipt of external information through the sensory system all the way up brainstem and spinal cord, how you have all of the processes that are going to act on that information before you come to any of your motor outputs. And those motor outputs are autonomic, therefore covering visceral output, as well as somatic and skeletal muscle and vascular smooth muscle, and so on. And we've talked about how you make use of regulating that with information that comes in and out of cerebellum, through the thalamus, up to cortex, cortical processing that allows you to take that information and make decisions about it, not just have knee jerks or jaw jerks or any other type of simple reflex, but to actually do complex things with it that range from building visual information into a sense of space around you, as much as trying to decide which of the things that you see in front of you is the appropriate object to pick up in the moment, and what sort of actions you need to do and what sort of planning you need to do, none of which is essentially simple reflex circuitry, okay? And the higher order functions require interaction and crosstalk between multiple regions, and those are going to be regulated by the thalamus, which essentially gates things as they come in, the basal ganglia, which act essentially as a sort of valve to turn things on and off through that complex balance between the direct and the indirect pathways that we typically teach as based on motor, but really spill out into all sorts of behavioral components. And then what we categorized as limbic, which is really combinations of thalamus, basal ganglia, and cortical-like structures that give this the content. You may be experiencing something in the moment, but in order to interpret it, you need to know, A, is this something I remember? That comes part from hippocampus for relatively short-term memory. Long-term memory, we're not really sure where it resides. And also, what does it mean? What is its emotional and valence and the like? And all of that comes out of limbic. So before I start to talk about how the diffuse systems interact with this, I wanted to remember to use the keyboard. And I want to sort of point out that there's some structures here that I haven't mentioned yet, some of which we will cover over the course of the morning. But here's a nice high-power section. Let's think about what we've got. So what is this? Corpus callosum. This? Lateral ventricle. This? Caudate. This? Anterior limb of the internal capsule, as specific as you can get. This? Putamen. This? Globus pallidus. What part? What's this? Yeah, anterior commissure. What's this? Yeah, we're probably a little bit behind the chiasm, just because what I've got is more bulge on the lateral than the coming to the midline. We're probably just at about the chiasmatic level. We're probably a little behind it. It's sort of the chiasm slash optic. What? Tract, yep. Okay. What's this? Yeah, parahippocampoid gyrus. Actually, it's really sort of uncus up here. What's this gray matter underneath it? So what do we know about hippocampus? It's a layered structure. Does this look layered? No. And we're a little anterior to the hippocampus, and we're a little superior to the hippocampus, so what might it be? Amygdala. All right? And what's going to be What's this? Yeah, fornix coming back. And as we will see this morning, this region here is medial septal nucleus, and in here we will find the basal forebrain. But what fibers are going to be making a loop like this? Yeah, the amygdalofugal fibers. Okay. What's this cortex? Insula. All right. Now, how about this gray matter here? Yeah, that's the claustrum. I haven't mentioned it before, but that's the last structure that I wanted to mention on this section. Now, let's look at it. We see a gray matter sheet. Okay. It's flat. And if it runs more, if you're present on more posterior sections, as you keep going back and it stays flat, you will have a sheet. And it's bounded by what? Yeah, so we've got white matter on this side, which is known as the external capsule. So you have an internal and an external. Nobody worried where the external was going to be when I kept talking about an internal capsule? Why couldn't you just call it capsule? Okay? The answer is because there's an external one. And then you've got white matter on the other side, which is interesting because it gets a name, which is extreme capsule. But what is it? Well, it's really the subcortical white matter of the insula. Okay? Any cortex has white matter right underneath it like that. What's in it? Axons. Great answer. Whose axons? Yeah, so remember, the insula has information coming in and information going out. And the white matter right underneath it is where those fibers are traveling. So the extreme capsule and the subcortical white matter of the insula are really different names for the same structure. So, sheet-like gray matter, and it sits between the external capsule that is up against the putamen and the extreme, which is up against the insula. Can you see it here? Yeah, probably, because I put the slide up, right? All right, so where do I find it? What's the easiest way to find it? Yeah, you start at the insula, and you go deep. You go through the subcortical white matter of the insula, and you come, which is also the extreme, and you come to the claustrum, external capsule. This is. Yeah, I heard it. Louder. Substantia nigra. Remember, that's substantia nigra, and then your red nucleus sits right here. Okay, this is peduncle, posterior limb, and then here we have genu. Look at this. People learned neuroanatomy this weekend. What do we have? Anterior nucleus. What do we have? Hippocampus. All right. What have we got right here? Optic tracts. Excellent. Callosum. Fornix. Notice that there's something between them, and it's not blue. Therefore, it's not white. We'll talk about that in about 45 minutes, and I've learned not to press this button. And here's the claustrum circled for you. So when we think about claustrum, it is a widely connected structure that is connected to cortex, okay? And it is widely connected. The size of it, it's a pretty small sheet of cells that is widely connected to cortex, which implies that there's very high convergence. You've got a lot more cortex surface area than you have claustrum. So you have to have lots of convergence. So what does convergence of cortical information usually mean? Well, it means that it's an output, but it means that you're actually not at the very beginning of processing. you're actually doing the higher complicated processing of doing all of the value assessment, all of the spatial assessment, knowing where you are, knowing context, and making decisions. It's really, in that sense, what you would think about as being a much more higher order structure. And it is reciprocally connected back in. There are a couple of striking examples. There's no connection to the primary auditory cortex. Where does the primary auditory cortex live? Yeah, Heschel's gyrus. And that is superior, posterior, temporal gyrus on that flat horizontal part that sits in, sliding in towards the posterior portion of the insula, right in the back. Okay? So, the claustrum is overplayed in many. Here's just a map of its connections back and forth, so you can see, here's a rough size of that thin sheet of cells, widely connected to most cortical areas, okay? Not notably the posterior superior portion of the temporal gyrus, and receiving input from all of them. And it's interesting, it gets it from visual processing areas, although not from auditory. People know who this is? Yeah, it's easier, maybe easier to recognize him here at the point when they won the Nobel Prize. That's Watson and Crick. This is Francis Crick. And so Crick, this is Crick's final publication. Okay? What is the function of the claustrum? Okay? It's often quoted as claiming that the claustrum is actually the seat of consciousness, rather than cortex being the seat of consciousness. If you actually go back and read the paper, it's a massive overinterpretation of what they actually say. The paper is actually far more sensible and conservative than that. And most of what they talk about, in fact, comes from cats. And so one can argue as to whether cats have consciousness at all, let alone whether it sits in the claustrum. All right. So when we think about this, our system of, most of what we've talked about is really point-to-point information transfer. We have a pretty good handle on it when we think about really simple modalities. How is it that you go from pressure on the skin with a sensor through a neuron in the dorsal root ganglia, it synapses in the dorsal, if it's pressure for pain, it synapses where? Substantia gelatinosa portion of the cord. Axons project across the midline, they project into the thalamus, they synapse on another neuron in the thalamus, that projects into cortex, and we told the cortex that there was pressure of a certain amount on a particular spot in the body, okay? That's the simple point-to-point neuronal transmission where we can understand the information. Beyond that, in the cortex, we sort of lose where that information is, and we don't understand what's happened to it anymore, but we know that it's happened in roughly the same way. An excitatory neuron whose activity is modulated by local inhibitory interneurons has moved the information to a next spot, and so on, okay? And eventually we get to the point where we drive an upper motor neuron that synapses on a lower motor neuron, and that lower motor neuron contracts a muscle, and I pull my hand back from whatever is crushing my fingertip, okay? We know that, and that's all a synapse to a synapse, and we view that as direct content. What we think about the diffuse systems for is something that overlays this entire structure and is going to provide modulation, not with information about this type of information, but about context, about alertness, consciousness, sleep, and so on. And I sort of anticipated this on Friday when we talked about the ventrolateral preoptic area. And I said this is the control center for sleep-wake, but it does it by driving a variety of brainstem structures that in turn control cortical awareness and activation. And we're going to now hit those brainstem structures because that's what they are. So when we think about diffuse systems, they are typically widespread projections that come from very small discrete sites. Okay? So when we think about it, we think about the inferior olive and its collection of neurons. They project where? Cerebellum. Let's be more specific. Any inferior olive will project to the contralateral cerebellum as climbing fibers, okay? So they are essentially going from them directly to Purkinje cells, okay? It's a pretty reasonable size structure in the brainstem, and it has a reasonable number of Purkinje cells. You have a lot of them. But these nuclei are smaller, and their targets, they will make more synapses than olivary neurons do, okay? Each of them will cover a much, much wider area. They, as I say, are involved in regulation of activity, sort of gain setting much more than specific information transfer. They are a lot about state rather than information. And by state, we mean things like attention, level of responsiveness to stimuli, sleep-wake cycles, and so on. And when you look at the transmitters, they are different than the transmitters we've been talking about. Two major transmitters we've discussed for neurons. GABA for our inhibitory neurons, mostly local with a few exceptions. Major exception of a GABAergic projection neuron is the, looks like this, dendrites in a flat plane. Purkinje cell, remember, Purkinje cells are inhibitory projection neurons. They project, they are GABAergic projection cells that project to the deep nuclei. Are the deep nuclei GABAergic? No, they are glutamatergic, okay? We talked about a couple of exceptions to GABA versus glutamate. What's the major other transmitter? It's not very common. It's still minor, but what's the major minor? Spinal cord interneurons, some of the interneurons involved in the respiratory control system. Glycine. Glycine's the other inhibitory neurotransmitter, okay? And the antagonist, the blocking drug for glycine is strychnine, and that's why strychnine is poisonous, all right? So here the transmitters are very different. Dopamine, norepinephrine, serotonin, acetylcholine. We've talked about acetylcholine a lot. Where else do we see acetylcholine used as a transmitter? Motor neurons. What type? Any output motor neuron. Doesn't matter if it's going to skeletal muscle or to an autonomic ganglia. They're all cholinergic, okay? The difference is what type of synapse they reach on the far side, nicotinic neuromuscular versus ganglionic, okay? And histamine. So I mentioned these, and this was a slide that I lifted from a lecture about a week and a half ago, which is inputs into cortex. Remember we talked about where things come in. The thalamic inputs typically come into layer 4 for the primary motor and sensory cortices. The input is in supragranular. What does supragranular mean? Above layer 4, okay? For the association cortices, whereas the diffusely projecting systems, which are typically the dopaminergic, cholinergic, noradrenergic, and serotonergic, are widespread. They are not transferring information to a specific input point in this cortex, but rather modulating cortical activity throughout. So we'll start with the catecholamines. These share a synthetic pathway, and they are derived from tyrosine, which has what is its ring structure is the catechol ring. For all of these, the first enzyme step is the rate-limiting step in the rates of production. So we use that as the marker for these cells. If you don't have enough of that, you can't be making enough of the transmitter. And you can also use transcription factors now to mark them because they have distinct transcription factors that turn on the repertoire. So if you're going to make a new transmitter, what do you need to do? You're a completely mutagenized neuron. You have made the new transmitter compound X. What do? What are the components you need to do to be a X neuron? Well, you need to synthesize it. So you're going to have some enzymes that you're going to need to synthesize it. What do you do with transmitters once you synthesize them? What? Well, well, either you make it down by the synapse or you have to transport it, but do you transport them as free-floating molecules? No, you got to get them into a synaptic vesicle component. So what are you going to need for that? some sort of transport mechanism that moves it from the cytoplasm where you make it into the vesicle, okay? And now you got vesicles, and you assume you're going to co-opt your prior vesicle release system, but then what do? Yeah, so you have to do something to do turnoff, and many of these use reuptake, okay? That's far more efficient. You can reuptake your transmitter. You don't have to make it again, right? That's pretty helpful, okay? So you have these whole constellations of transcriptional profiles that include synthetic pathways, vesicular packaging pathways, and uptake pathways. And guess what? They're all controlled by distinct profiles of transcription factors. So nowadays we can use transcription factors to mark these. But that wasn't always true. We used to use enzymes, and we still do. But in the old days, we did really crude things. So this is Falk and Hillarp. If you read old neuroanatomic and physiology papers, you will come across this method. They basically took sections, and what they discovered was that one of them was a chemist. I don't remember which. If you took these compounds, the catechol nuclei, that were different between tyrosine and dopamine and dopa and epinephrine and norepinephrine, they're all slightly different, and you make an adduct with formaldehyde, they will fluoresce, and they will fluoresce slightly different colors. So if you have a way of making that reaction in your tissue, you can see different colors for different transmitters, and you can map them. And this is, in fact, the way these were mapped. They were not mapped by immunohistochemistry, because there weren't good monoclonal antibodies in the 60s when these were being done, and they definitely weren't mapped by transcription factors, because we didn't even have the word transcription factor when these were being done. They were mapped by this autofluorescence. Having done the method, let me tell you how it's done. You cut your sections, frozen sections, you put them into a 60-degree oven, and you put an big open beaker of formaldehyde in the bottom, with the goal of the oven filling with formaldehyde vapor that then does the adduct of the catechols. Unfortunately, your entire lab fills pervasively with formaldehyde vapor, blinding you, making you completely stink of formaldehyde. However much you might think you smelled of formalin coming out of the anatomy lab, it has nothing to do with this. When we would do this reaction, we would like tape the door closed, put on masks. It was horrible. It's not really done anymore for that reason, but it is actually effective. And it was what was used to do all of the anatomy that I'm going to talk to you about the catechol system, and in fact, because of other adducts, the serotonergic system. So here's tyrosine, okay? And we begin with our rate-limiting enzyme, which is tyrosine hydroxylase, okay? Adds this extra hydroxyl group. This is dopa, okay? Dopa is acted on by an enzyme that is typically called dopa decarboxylase, but it's really got far broader specificity. It acts on all aromatic amino acids, and it decarboxylates it out here. So this carboxyl group comes off. And then we have dopamine, okay? So dopamine is the first transmitter we're going to talk about. But if you are not a dopaminergic neuron, so what enzyme would you use to mark yourself as a dopaminergic neuron? You would use tyrosine hydroxylase. You wouldn't use dopa decarboxylase because it's not actually specific for the dopamine pathway. It's actually an aromatic. Whereas what is specific for this pathway is tyrosine hydroxylase, okay? But if you're trying to make norepinephrine, you will then use dopamine beta hydroxylase to put one more hydroxyl group on the beta sugar, the beta carbon here, and make norepinephrine, okay? So in this pathway, if you stain for tyrosine hydroxylase, what neurons will you identify? Dopaminergic, and noradrenergic, and adrenergic. We haven't gotten to them yet. Dopamine beta hydroxylase will tell you the difference between dopaminergic and noradrenergic. And then to go from norepinephrine, because I couldn't squeeze it into the figure and have everything be legible, it's phenylethanolamine and methyltransferase that puts the last methyl group back on and makes epinephrine. There aren't that many adrenergic, epinephrinergic neurons in CNS. There are some. Most of this is going to happen where? Adrenal. It's a bit more specific. Medulla. Okay? Okay, remember the cortex, glomerulosa, fasciculata, reticulata, GFR. Okay, that's about all I remember. Okay. All right, so the distinct nuclei are marked when we first did it by the autofluorescence. They run essentially from the brainstem up to the hypothalamus. Okay? So that's the zone in which we'll see them. The higher up they are, the more likely they are to be dopaminergic, and the lower down they are, the more likely they are to be adrenergic, noradrenergic, and then eventually adrenergic. Okay? So in fact, we see a gradient of the transcription factor expression the same way we see lots of other transcription factor patterning gradients. And in fact, here listed from the bottom in the medulla all the way up through hypothalamus, we have the nuclei, and they are known as groups A and a number. And some of these we know about already. Remember, we know about, what do we know about the dopaminergic nuclei in the hypothalamus? What do they control? Yeah, they are in what type of regulator of prolactin? Negative regulators of prolactin. So we knew about them already. Gee, you don't have to learn them today. Okay? We already know about this one. What does that do? Yeah, that's the nigrostriatal pathway. That's what regulates our balance of direct and indirect pathways. Now, in fact, we will see that there are comparable other midbrain nuclei that do that. And then the ones that we really are going to care about are locus ceruleus, which is the major noradrenergic nucleus in the pons. And look here. Dorsal motor nucleus of the vagus has some adrenergic components. Interesting. It's not only the parasympathetic, it has some other projections that we've talked about. But the ones that we really are going to care about here are going to be the locus, the nigra, the VTA, and to a lesser extent the retrorubral tegmental nucleus. Okay? But the gradient is such that the lower down you are, adrenergic, noradrenergic, higher up, dopaminergic. All right, so let's start pretty high up in the brainstem. And we are here, where? Midbrain. Okay, and we can see the nigra. And where would you find the ventral tegmental area? In the ventral tegmentum. Okay? So when we think about tegmentum in the midbrain, what are we using it to distinguish between? Well, dorsal and ventral is the gradient within it, but what's the sort of dichotomous separation when we talk about parts of midbrain? Well, let me frame it this way. When I draw the basis pontus, like this, okay? And here's my nice basilar artery here, and this is my basis pontus with bundles. What are these bundles? Yeah, and what are, those are pontocerebellar fibers. What are these? Corticospinal, and what else is in here? Well, some cortical bulbars, but what else in here is besides white matter? Pontinuclei, so that these neurons that are in here, okay? But when we think about that, we have this is basis pontus, and this is pontine tegmentum, right? Okay? That's the dichotomous separation we make there, okay? How about in the midbrain? What's the split we make? Tectum versus tegmentum. So essentially anything that is not the peduncles and not the tectum is here. So this is going to be our tectal plate. This is all going to be our tegmentum. And the ventral tegmentum is going to be down at this end, and down here is where we will have the ventral tegmental area. It is not physically separable from the nigra. It's only sort of by relative location. If you show me a picture of a VTA neuron, I cannot tell you that it is not a nigral neuron. They look identical, okay? It is only about location. And then retrorubral field should be on the other side of what? Red nucleus, okay? Its name tells you where it lives. It's on the other side. It's separated from the other large dopaminergic neurons in the midbrain. So that's that boundary, and it actually lines up almost perfectly with where I drew the blue line, okay? It's a relative point. The nigra tends, if you get a thick enough section, you can see it sort of bend, and it's that bent region here. Here's the interpeduncular nucleus. Anybody remember who projects to the interpeduncular nucleus? The habenula via the white matter bundle that bends backwards. Yeah, fasciculus retroflexus. That's right there, and that's wired much more like hypothalamus than like thalamus, even though the habenula sits on the surface of the thalamus. So where are these? Well, this is nigra coming down. This area here would be likely to be VTA. In here's where we will find interpeduncular. And where will we find the retrorubral? Where's the red nucleus? Here I am in the aqueduct. Tell me how to find the left retrorubral. Anterior. Yeah, let's go ventral. Let's go ventral. Tell me what to do, somebody. Yeah, I don't know why that line appeared, but it's basically, okay, so this is red, okay? What's this white matter? What are these white matter bundles here? Yeah, those are fibers of 3. What's this? MLF, okay? And so the retrorubral field will be right over here. It's not actually that far from the nigra. What's in between it here? Medial lemniscus, spinal thalamics. Okay. When you look at it on the gross, it's exactly the same thing. Here is the nigra coming in, and the region down here will be VTA. And then here's that beginnings of the red with some fibers of the superior cerebellar peduncle around it, and the retrorubral field will be right there. Yep. Here, we're not, it would be in this rough zone, but you can't see it as a discrete structure. So at this level, we probably have got, just trying to see, those are fibers of 4, so this is lateral lemniscus out here, because this is coming into inferior colliculus. If we were above the inferior colliculus, it would likely be the brachium of the inferior colliculus, which are the inferior collicular projections to the medial geniculate. All right. So when we think about that ventral tegmental area, it has wider spread projections than the nigra does. Nigra is much, much bigger than the VTA, but where does the nigra go? Nigrostriatum. So where does it go? Striatum. Okay? It's a relatively small target. It's an important one, but it's relatively small. Whereas the VTA goes to cortex, and it does not go all the way to the occipital pole. It tends to come to frontal, cingulate, and back into the early bits of parietal lobe. It comes down into hypothalamus, amygdala, and hippocampus. So those have dopaminergic innervations. When we think about those that come, and here we can see the nigra, SNC, okay, has this incredibly dense projection into striatum. And it goes all the way around. What's going on here? What's this back here? That's the tail. It's actually going to loop around and come even more anterior. Remember that caudate, all of that has got dopaminergic innervation, and all of it comes from the nigra. Whereas we have this innervation that's going to come to the cortex and out and around, mostly through frontal and into cingulate, from the VTA. Okay? It also projects back here. Little bump on the back of the thalamus, abbreviated HB, that would be the habenula. Okay, so when we sort these out, and I've given you their A names, because sometimes in papers people talk about them that way, although here they tend to use the neuroanatomic. When we think about the nigra, it is striatal, predominantly caudate and putamen. What's the other component of striatum that we haven't really talked about yet? Accumbens. Okay? In contrast, the VTA has some striatal targets, particularly the accumbens. And that striatal accumbens component is critically important. It comes into limbic, mostly amygdala, goes to prefrontal, insula, and cingulate. It talks into thalamus and hypothalamus, and it actually goes down all the way as far as the medulla. Okay? Hippocampus, okay, is mostly covered by retrorubral. So we've got some dichotomy. Hippocampus and amygdala are split here, between these, okay? Striatum is split between these, okay? And cortex is almost exclusively covered by the VTA. Okay? And these are all going to target dopaminergic receptors. But remember, those can be incredibly complex. There are multiple types, but we categorize them into sort of the D1 family and the D2 family, okay? Which are either excitatory or inhibitory. And the goal, if we come back here, is that D1s are excitatory, so that the dopaminergic nigrostriatal allows that direct pathway to go and inhibits the indirect. Okay? And when you turn it off, you end up with the indirect pathway, which decreases motor activity, overwhelming any direct to allow your motor to component. This is just the same slide that I showed when we talked about basal ganglia a week ago. Okay. So the VTA, let's think about what it does. It's got these unique targets, including the accumbens, amygdala, cingulate. We haven't talked about the accumbens yet, but what do we know about prefrontal cortex and amygdala? What are they really involved in? What type of processing? Specific sensory modality? No. Motor output? No. I mean, they're really involved in what? Judgment, decision-making, value assessment, okay? So here's a dopaminergic system that is built to balance and influence those processes, okay? So it's clearly, in fact, as we will see later on, important for both detection and satisfaction of reward, and you'll hear a whole lecture at the end of the course on reward mechanisms, but also on that decision-making process. All right, so let's slip down into the midbrain, into the pons rather, and at this point we're going to shift away from dopaminergic neurons to noradrenergic neurons, okay? So groups A1 through A7, 8 is where we began in the midbrain, and the ones that we really care the most about are the locus coeruleus, and then to a much lesser extent the lateral medullary reticular formation. But we're going to focus on locus much, much more. So where will we find the locus? You probably have seen it on the brains in lab, I hope. It is, what does locus coeruleus mean? The blue spot. It's not as blue as any of us would call something, but it is easily recognized in people who don't have underlying either Parkinson's disease or Alzheimer's disease, both of which lead to neuronal loss from the locus and depigmentation. So it's easy to see, and where does it sit? Well, let's think about what we have. These are corticospinal bundles. The gray matter is pontine nuclei. The white matter bundles are, so pontocerebellar fibers. Okay, this is our medial lemniscus. This is superior what? Cerebellar peduncle. This is MLF. This is, top end of the fourth. So where will we find the locus? Anybody remember the dot from seeing it on the brains in lab? Find it right over here. And it's in fact this collection of neurons right there and here. There's the locus. It's about that big. It is a dot. It's tiny compared to the nigra. It's actually a column of neurons. It's a round column. Tapers as you come to the two ends. But it's pretty small, and it's located lateral portion of the beginning of the periaqueductal gray matter at the upper end of the fourth. Here it is on the gross. And in fact, if you look, I hope you can see that as you go in here, you can actually see it through the ependymal surface here. So it's this column of neurons. Okay? It's actually a cylinder like this. Okay? That's our locus. Okay? Not very big. Well, that is enormously widely projecting. Okay? So it covers all of cortex, unlike the dopaminergic system, which doesn't reach the occipital lobe. It goes through all of thalamus. It goes through hypothalamus. It goes to amygdala. It goes to hippocampus. It goes to cerebellum. It goes to the brainstem, and it goes all the way down to the cord. Okay? That collection of neurons project far more widely than just about any other in the nervous system. Okay? Now, when we think about it, it's a column, and in fact, the higher up you are in that column, what type of targets would you expect to see? Higher targets, exactly. So that the upper end of that is your cortical. As you come down, you begin to come into neurons that project towards thalamus, amygdala, hippocampus, and so on. And then as you come, and that's the list of targets that are here, but it's really all of the structures that we've covered so far. And then as you go to the lower end of the locus, you are mostly downwardly directed. Okay? So you come down and you regulate those sensory nuclei for processing. You regulate the output nuclei, and you regulate, in fact, basis pontis. What is regulating basis pontis nuclei going to do? You shut them down. What are you taking out? What loop are you disrupting? So what drives the basis pontis nuclei? Cortex, corticopontine fibers. Those what do the pontine nuclei drive? Cerebellum. Where does that output go? Most of it's about extremities, so where is it going to go? Back across the midline to the red nucleus, thalamus, thalamus to motor cortex. So what are you doing if you inhibit that system by shutting down your adrenergics, noradrenergics? Quieting motor activity out. When you turn it on and gate it, you're allowing motor activity to flow. So these are all associated with increased activity, typically with so-called wakefulness, arousal, attention, vigilance. The ability to pay attention to subjects is controlled in part by which sets of neurons in the locus drive what parts of structures. They increase activity in response to pain. What does that tell you? They have to have sensory or somehow sensory or processed information has to come back into them. Okay? Where do you think that's going to come from? Going to come actually mostly from thalamus, and thalamus via amygdala, and amygdala via hypothalamus, down to control the brainstem. Okay? And the lower ones tend to project down, and they project all the way down into cord. All right. So those are the only ones that are actually made from the catecholamine nucleus. First by tyrosine hydroxylase, and then by dopamine beta hydroxylase. Okay? So let me just say a quick word. So if people know the major pharmacologic therapy for Parkinson's disease? Dopa, L-Dopa. So why do you give L-Dopa? Why don't you just give dopamine? Yeah, it doesn't cross blood-brain barriers. So you can get L-Dopa in, and it turns out you don't actually give just L-Dopa. So the standard medication is, anybody know? So it's a combination of L-Dopa plus carbidopa. How come? So why you give something that doesn't cross the blood-brain barrier, carbidopa? Well, what if you have dopa decarboxylase in the periphery? What's going to happen when you give somebody L-dopa? You make dopamine in the periphery. Do you want that? No, you want that dopamine to be able to get in. So instead, you basically provide another substrate for the peripheral dopa decarboxylase to allow it to work on that. So it for which it has a higher affinity, so that your L-dopa is able to traverse. What happens in the nervous system when you give it L-dopa? So what's the rate-limiting enzymatic step for making dopamine? Tyrosine hydroxylase. Ah, are you behind that? You're past that. So now you're providing cells with L-dopa. They can take it up, and what have they got lots of? Dopa decarboxylase. So what do? They make more dopamine. So the way that therapy works is it relies on existing dopaminergic neurons that you basically overload. So instead of having 100 neurons releasing, if you're down to only 50 neurons, each one of them will release more. So you get more dopamine to your nigrostriatal target. Okay? What's the consequence of that mode of therapy? What's going to happen in a progressive disease? Eventually it loses its efficacy because you don't have enough dopaminergic neurons left to load. It requires having a dopaminergic neuron there to stuff with L-dopa to overload it with dopamine so that it still has a function. If the neurons are all gone, you can give as much L-dopa as you want and it won't give you any benefit. And that's exactly what happens over time. So let's think about serotonin. So it's made from tryptophan. So it's not really a catechol, but it often is grouped together. Okay? And it's done in basically the exact parallel mechanism. Okay? So here we have tryptophan hydroxylase. Then we have a decarboxylase. This is an aromatic amino acid decarboxylase. It has in some cells it's the same as dopa decarboxylase. And you make serotonin. How do you get rid of serotonin? So reuptake. Selective serotonin reuptake inhibitors are therapeutic because what are they going to do? What's their physiologic consequence going to be? Yeah, they're going to increase facilitating serotonergic transmission. You basically keep it in the synaptic cleft longer. Okay? What's the other way to get rid of these? Monoamine oxidase. Okay? And that converts it to 5-hydroxyindole acetic acid, which is a metabolite you can actually measure. Okay? So you can measure that as a way of determining serotonergic levels. So again, here, tryptophan hydroxylase is the rate-limiting enzyme. And these nuclei are in the raphe. And I mentioned the raphe in the second lecture. Okay? I said these are midline structures, mostly in pons and medulla, that are widely projecting. And here they are. There's a little bit of them up in the midbrain, big raphe in the upper pons, and then they continue down into the medulla. Notice these are numbered with the B. Okay? So our A's were our dopaminergic, noradrenergic, and adrenergic, and the serotonergics are B. Okay? So if you read nomenclature, that's what they are. And so where will the raphe be? Here's my arrow, and here's my dot in the ventricle. Where do I go? Ventral. Stop. And they actually are right on either side here. They are not that little midline seam, but they are right on either side. So they're even smaller. So you can't see them. I know that they are here, sitting right in here and here. And that's the dorsal raphe, as we call it. And it's sometimes called the magnocellular dorsal raphe. It's really big neurons, and they project really, really widely. So that raphe system has the same spatial organization as we talked about for locus. Except instead of being a short column, we've now got a column that really runs the whole brainstem. Higher up you are, the higher you go. And it goes everywhere. Ball of cortex, thalamus, amygdala, hypothalamus, hippocampus, comes down, it goes into cerebellum. And then as you come to the lower ones, in the lower pons and in the medulla, these are going to project downwards into other brainstem nuclei and down into the spinal cord. Okay? This one, too, is really important for keeping you awake for arousal and attention. The issue here is that if you thought dopaminergic receptors were heterogeneous, serotonergic receptors are even more heterogeneous. Okay? It's a massive collection of receptors. It has both G protein linked and ion channels. But the two things to remember are that there are two major categories of drug that we know about that affect the system. One is the selective serotonin reuptake inhibitors. Okay? And they work to basically be system agonists by prolonging dwell time in the synaptic cleft. The other is LSD, okay, which is a complex, weak agonist at some and antagonist at others. And it is a massively potent hallucinogenic drug, in part because it is probably stimulating some parts of serotonergic systems and making cortex be hyperactive, generating the hallucinations, at the same time quieting other areas so that the context for them is lost, so that it's a very complex function. It's not a single on or off type target. And the serotonergic system is important for both arousal and attention and all sorts of behavioral functions. All right. Anybody here ever take an old style, apart from like me and the other, take an old style antihistamine like clotrimazole? Okay, so in the old, people here have allergies and takes, some form of regular antihistamine. Yeah, okay. In the old days, the only antihistamines that were available were not receptor subtype specific, okay? And once you know that and you had the experience of taking them, you would know the physiologic function of the histaminergic projection, which is drugs like clotrimazole were massively sedating, okay? You'd get your dose in the morning and you'd fall asleep walking to school. I mean, they're massively sedating because the histaminergic system works to keep you awake. All of these systems are keeping you awake, okay? They're all about enhancing alertness, arousal, and wakefulness, okay? Now, what you now take is a 5-HT3 blocker, which works out in the periphery. There are none of those receptors in the CNS. The drugs don't cross the CNS. They don't work on any of the CNS receptor subtypes, and therefore you get only the blockade of the immune system's histaminergic components, and you can go about your business without your allergic symptoms. But within CNS, these are really important for arousal. There is one tiny little nucleus, tuberomammillary. Where would that be? Yeah, it's some, so where's tuber? Yeah, hypothalamus. Where's mammillary body? Hypothalamus. So where should the tuberomammillary nucleus be? Hypothalamus. And roughly where is the tuber? What is the other name for the tuber? The infundibulum, the stalk of the pituitary. Where in the hypothalamus is that? Let's think about our hypothalamic anatomy again. When we take the hypothalamus, the anterior border of the hypothalamus is going to be the anterior wall of the third ventricle, but what's the sort of physical structure we would think about? Lamina terminalis, and right behind it we will come to the chiasm, right? Posterior boundary, mammillary bodies. In between them, we've got the hypothalamic stalk. So when we think about our hypothalamus, we've got the midline here. We have the boundaries that separate medial and lateral, and what are they? We've got the fornix and mammillothalamic. So we've got our lateral zone, which is all out here. And we have our medial zone, and remember we divide our medial zone up into roughly thirds. We've got that chiasmatic region, we've got the mammillary region, and we have the tuberal region in the middle. Okay? So the tubero-mammillary nucleus is going to sit right about there. Okay? If you think about its name, you think about the names you already know, you know exactly where it has to sit. And that is where the histaminergic projections come from. So these go most everywhere. Okay? So really, as you think about these, noradrenergic is everywhere, serotonergic is everywhere, histaminergic is everywhere, dopaminergic is the exception. Okay? Even when it goes to many places, there's very little descending brainstem and core dopaminergic innervation, and not even all the cortex gets dopaminergic innervation, but really the vast bulk of it is focused where? Striatum. Whether it comes from nigra to caudate and putamen, or VTA to accumbens, that's the major dopaminergic target. All right. Now, all of these, the nigra, yep. Right. Everywhere. So, in humans, we have absolutely no idea. And for the histaminergic, we don't understand its inputs and outputs particularly well. It's not particularly well studied. We know that the best one that's known are the differences between nigra and VTA, which function in very different contexts and are wired very differently. The assumption here is that if you wish to turn the system on, you probably are turning it all on. Now, whether at every moment you are gating every piece equally is hard to know. Now, I showed this slide. Yep.
Are these neurons firing serotonin and?
No. So these are just purely, the tuberomammillary are just histaminergic. Okay? So each of these is an exclusive. Okay? They do one of them. All the systems together coordinate. And as I showed you, this is a slide from Friday. This is the ventrolateral preoptic area. Remember, when I showed you that balance, I showed it controlling the locus. So that the ventrolateral preoptic, which is right next to the suprachiasmatic, okay, and gets your information about rhythmicity, drives all of these brainstem nuclei. Locus, tuberomammillary. We'll come to the basal forebrain in a bit. But this is essentially the way the circuit works. This is the driver that controls these, and when these are turned on, they project everywhere and say, oh, wake up. Okay? And when they are removed from the inhibition, that's when they are removed from the inhibition from the ventrolateral preoptic. When the ventrolateral preoptic is firing, it is telling these to stop firing, and you become asleep. Okay? So this is the mechanism by which that tiny area affects everything, is it goes to the known widespread circuits. All right, let's take 10 minutes and pick up again at 20 of for both acetylcholine and then bits about accumbens and extended amygdala. All right, so we've covered our adrenergic, noradrenergic, dopaminergic, and those nuclei were labeled with A's. We covered our serotonergic, and those nuclei were B. The histaminergics weren't discovered. Any idea why? They were all discovered using the adducts with formaldehyde vapor, and it turns out that you don't get a color fluorescence with histamine, so nobody recognized them until significantly later. Okay? But the last group that was recognized because people knew how to stain for both the synthetic enzyme or to look for the activity of degrading acetylcholine. So they were histochemical reactions for acetylcholinesterase, so you could find cholinergic projections. and these are the cholinergic groups. And we basically have 4 major groups. And we need to remember that these are cholinergic nuclei that are different from our lower motor neurons and our intermediolateral cell column, which are also cholinergic, okay? As well as our dorsomotor nucleus of the vagus. What else? Other brainstem cholinergic nuclei. Yeah. Edinger Westphal. Other cholinergic groups. You're going to make me cry. Cry. Yes, superior salvatory and inferior salvatory. Okay? These are all cholinergic, but they are not what we're talking about now. Why not? What are the targets for all of these? Either muscle or ganglia. Okay? So those are all projecting out of the central nervous system. These that we care about and talking about now are all staying within central nervous system. These are not taking information to nicotinic neuromuscular or nicotinic ganglionic receptors. But rather, and these all sit in the basal forebrain. Where would I find the basal forebrain? It would be on the basal surface of something. And when we say forebrain, what do we mean? Well, where's hind? I haven't used these terms, but where would you think the hindbrain would be? Cerebellum, sort of in the back, right? So where would forebrain be? Up in the front. Yeah. So basal forebrain is on the basal surface of the cerebral hemisphere. Okay? And we will find the medial septal nucleus. We already know about something that projects to the medial septal nucleus, which is so we have our hippocampus, which is a long sausage, on the surface of which we have white matter fibers that are the alveus that are running up and turning and joining the fimbria, that is growing and growing and growing until we get to the posterior end of the hippocampus. We have the fimbria, and what does it do? It fimbrillates. What does it do? Becomes the fornix and comes forward. And what does it do at this point when it has come to the anterior commissure? Splits. And we have postcommissural fibers that are going where? Mammillary body. Mammillary body projects where? Anterior nucleus of the thalamus. Through the mammillothalamic tract. Anterior nucleus of the thalamus projects to the cingulate. Okay? Great. But how about those fibers that came in front of the fornix, the precommissural fibers? Where did we say they went? Medial septal nucleus. Ah, so that tells us where the medial septal region is. Right in front of the anterior commissure in the brain there. Okay? So when we think about it, we know where that is. Nucleus basalis is going to be oriented like the basal surface. And so we've got something here that's going to be oriented in this plane. And nucleus basalis is going to be oriented like that. And then we've got something called the diagonal band of Broca that's going to connect them. Okay? Nomenclature makes sense. Okay? You can figure all of a neuroanatomy out on your own. So this is our medial septal. Oop. T goes on the other side. Don't try and read. This is why there are no fill-in-the-blank questions on the exams, because none of us can figure out how to interpret your misspellings. You can't read mine. This is the diagonal band of Broca, and this will be the nucleus basalis of Meinert, as we will come to. All right. So we've got these. So let's see where they are. Meinert. This is Meinert. He was a student of Rokitansky. Rokitansky? Anybody know who Rokitansky was? So Rokitansky was the other father of pathology besides Virchow. Okay, those of you who did an autopsy last year as part of PATH, where they took the block out, remember that? Man in a pan, okay? Out it came. That's the Rokitansky method. The Virchow autopsy method is dissecting everything in situ. Meinert was the head, in fact, of the psychiatric clinic, okay? His students, recognize a few names, okay? Freud we haven't talked about. He did actually do some neuroanatomy. We've not talked about his contributions. Forel. What is Forel known for? The fields of Forel. Yes, that picture of the Institute of Psychiatry in Zurich. You will never forget the fields of Forel. Okay? Wernicke and Korsakoff, who came in the opposite order for which their syndrome is named. And Meinert actually really focused on neuroanatomy underlying psychiatric disease, interestingly. So where are these structures? Are we in front or behind the anterior commissure? What's your clue? So what I would say is let's use this as our clue. What is it? Yeah, it's actually labeled for you on the other side in an abbreviation. So it's the optic chiasm. So is the optic chiasm in front or behind of the anterior commissure? Think about the midline view. I don't have one pulled up here. It's in a different lecture. But is it in front, anterior, or posterior to the anterior commissure? The optic chiasm is anterior to the anterior commissure. So if we see the optic chiasm, are we in front of the anterior commissure? Yes. So we are, in fact, looking at the septal nuclei in here where the precommissural fibers will come. Okay? And you know that because you know what the midline structures look like. He said confidently. So here is our anterior medial septal, and then the second one is the diagonal band of Broca that begins to angle. Now where are we? Well, yeah, we move posterior, and what do we see? Right here? Anterior commissure. What is this? Optic tract. So if you're at the optic tract, at the anterior commissure, you'll see optic tract, not optic chiasm. So we are now at the point where you can see the fornix coming right down, and most of these fibers are going to go where? Mammillary body. Far more of them are post-commissural than pre-commissural. But now what's this? Let's start over here. This is GPE. Okay, this is anterior commissure, right. Remember, it continues like that, and it's eventually heading out to the temporal stem over here. And this region down here is what we would call basal forebrain. It's on the basal surface of the forebrain. And in here is where we will find the nucleus basalis of Meinert, right in here. Okay. Take a look at this zone right here. See that? I don't like the way this atlas identifies it. I think they're wrong. If you look here, before I drew all over it, it looked a lot like pallidum. In fact, if you looked at the texture of it, on your copies, it looks like the pallidum. And as we'll get to in a few minutes, that is in fact ventral pallidum. And ventral pallidum is a part of the pallidum that is outside the ice cream cone. I'm sorry, there's nothing I can do. My rules don't hold perfectly. So here we've got a little bit of septal nuclei, and now running horizontally, we have the nucleus basalis. So the, yep?
I have a student oriented question. Is there any, for me, the optic chiasm and tract look exactly the same? Is it just the anterior commissure that you're using to distinguish?
Well, so they are both white matter, but when you come, the optic chiasm is essentially a midline structure, and it has, as you look at it, as you come all the way to the midline, you've got nothing but white matter. The optic tracts are two a left and a right. They're on the other side, and they will have hypothalamus between them. And so when you look here, you can see hypothalamus between, coming down here. Okay? So the way I like to think about these, if you begin anteriorly, here are your medial septal nuclei. As you move posteriorly, this band of cholinergic neurons angles, diagonal band of Broca. There is no structure that can't be split into two, so the diagonal band of Broca has what's called a vertical limb and a horizontal limb. Don't worry about it. And then as you move further posteriorly, this sheet spreads out and is now flat in this sort of basal plane, and that's the nucleus basalis of Minor. A continuous sheet of cholinergic cells. That's impossible for me to draw in any way, nor have I found any useful illustration of it. I can only do that with my hands. So when we think about them, we've got what we call medial septal, nucleus of the diagonal band. This is the horizontal, and that's the vertical, and that's the horizontal limb. We group them together. And the nucleus basalis. And they make this sort of L-shaped flat sheet that runs posteriorly. So that Minor extends far more posteriorly than the septal region. It's way underneath the anterior commissure and behind it. Whereas the septal nucleus, we know, is way in front of the anterior commissure, because that's where those precommissural fibers are headed. So where do they go? Well, medial septal goes to the habenula and the ventral tegmental area. So we have cholinergic talking to dopaminergic. Medial septal goes to the hippocampus. Diagonal band does too, but it's mostly medial septal. To amygdala and cortex is nucleus basalis of Minor. Okay? So nucleus basalis is our cortical cholinergic innervation. Septal is the hippocampal cholinergic innervation. Okay? And amygdala, for this situation, travels with cortex. Nucleus basalis is important because it's affected early in several diseases. Okay? In both Alzheimer and Parkinson's disease, neurons here get the characteristic proteinaceous aggregated inclusions that mark the disease, tangles in Alzheimer's disease, Lewy bodies in Parkinson's disease. They also die. You have fewer neurons in that setting. If you lose those neurons, what are you going to lose? Cholinergic innervation of the cortex. Well, maybe that's what contributes to cognitive dysfunction in those diseases. So people gave drugs that blocked them, or gave drugs that enhanced cholinergic transmission. This is if I give somebody with nigral neuronal degeneration dopamine and make more dopamine, and they get benefit from that, what if I make them have more benefit from their cholinergic transmission? It's hard to really load things and make more acetylcholine. How come? Well, because the components that you use to make it are in such abundance, you can't load more, okay? You really just can't get more acetyl-CoA and choline in there. You can't do that, right? Because they're just so abundant because they're used for so much else. So that doesn't work. So what do you do instead? Yeah, you block the inactivation mechanism. And the inactivation mechanism for acetylcholine is acetylcholinesterase. There's not a reuptake system for it. It's not like serotonergic or dopaminergic systems or GABAergic systems or glutamatergic systems where what you do is reuptake and recycle. Here you degrade it with acetylcholinesterase. So what if you give a drug that is a acetylcholinesterase inhibitor? And these, in fact, have been tried, and they do show mild symptomatic benefit. And this was initially the cholinergic theory of Alzheimer's disease, and it was assumed that this was going to be the key to giving benefit. Turns out that if you actually test these people, they get benefit for about the first 6 months. But after 6 months, they catch up in deficits to people who didn't get treated, although they are almost always maintained on them forever. Mostly because they are relatively harmless. At the same time, yep. not significant. But the other thing that you should know is that the drug scopolamine, which is used as an antiemetic, anti-nausea, can give people amnestic syndrome. Why? It's a muscarinic blocker. So the assumption is that you are blocking that cholinergic transmission and you are disrupting memory. And we don't know whether that is in fact memory at the hippocampal level or at the cortical encoding level. But it does do it, and it's a known side effect. Not very common, but it can happen. So I just want to say a few words about septi. We've talked about the septal nuclei. We need to separate that. They are gray matter. They are bilateral. And because they're gray matter, they have inputs and outputs. The septum pellucidum is that glial boundary between the lateral ventricles. Okay? It's glial, has no neurons. There's one of them in almost everybody. Sometimes they're generated by fusion of two leaflets during development. Sometimes the two leaflets don't fuse, and it's known as a septum pellucidum. And you will see a lateral, normally if I were to draw it, I would draw a lateral ventricle to the septum, and like this with the caudate here. Okay? And then I would draw the lateral ventricle on the other side with a caudate here and my corpus callosum here. Sometimes instead of one membrane here, you can have two and a little bit of CSF filled space in between them. Completely benign variant. Okay? But the septum pellucidum has no neurons, no projections, no myelinated fibers. So here we can see this would be septum pellucidum. This would be septal nuclei in here and here. And then as we were to move further back, now what have we got? This is septum pellucidum. This is anterior commissure. So right in here we will find nucleus basalis. This is optic tracts. This is third. This will be hypothalamus, putamen, caudate, GPE, claustrum, insula, corpus callosum, amygdala right there. Okay. So, connections of the septal nuclei. We know that it gets a projection from the hippocampus, right? But what else did I tell you it does? It's cholinergic innervation of the hippocampus. Remember? So, what does that mean? How do you get into the hippocampus? Well, if you're not the perforant path, you actually come in via the fornix. So even though I told you that the fornix was essentially unidirectional, I lied. Okay? These fibers come backwards through the fornix. Okay? But that's what's going on. That's where your cholinergic gets there. Okay? They go to the amygdala, again by looping around via the stria terminalis, and the ventral tegmental area, and for that they use the median forebrain bundle. Okay? Remember, we talked about that coming in and out of the amygdala and traveling down. There are fibers that go to the habenula, because the habenula is a lot like hypothalamus, and there's input from the interpeduncular. So what do we have there? So we have septal to the habenula. We have interpedunc to the septal. And what have I forgotten to draw in? Fasciculus retroflexus. So we actually have another one of these three neuronal or three structural loops comparable to what we think about for Mollaret's triangle, although this one doesn't have anybody's name on it yet. It's probably too late. So the other thing that you need to know to be a well-educated adult about the septal nucleus is this paper, and then the subsequent series of studies that came from it. So this is a paper from 1954. Yes, things from 1954 are important. It's from Olds and Milner. This is a different Milner than the Brenda Milner who was involved in the first studies of HM, although both were in Montreal. These people were at McGill. Brenda Milner was at the Montreal Neurologic Institute. And this is a paper entitled Positive Reinforcement Produced by Electrical Stimulation of the Septal Area and Other Regions of the Rat Brain. And there is the lateral plane film of the rat brain with its rather crudely manufactured electrodes shoved into it. And then next to it is the cross-section of the rat brain showing where that particular electrode went. And it comes right in here, lateral to the midline. And in fact, the tip of the electrode, as that reconstructed it, was down around here. And what's that? Septal nucleus. Okay. So when the experiments were first done, what Olds was doing was basically, depending on where he saw the rat in the cage, stimulating. So when the rat was in one particular corner, he would stimulate the electrode. And what he observed was that the rat started to spend more and more time in that corner of the cage. Okay? Seemed to like it. So then what was done was to do other rats, and they've gradually figured out where they were supposed to be putting the electrodes to see the phenomena. And then they actually did the animal in a Skinner box. People know what a Skinner box is? Yeah, so it's essentially a operant training device actually, Skinner supposedly used one on his child as she grew up, but mostly used for animals these days, where you put the animal in and there's a lever and they get whatever the reward is if they press the lever in response to stimulus. And what was observed here was that if the lever was wired to stimulating the electrode, the rat would sit there and just go like this all day long. Wouldn't eat, wouldn't do anything else. It would just sit and tap the electrode for stimulation. Now, I was always taught, and I have actually no idea whether this is really true or not in the end, is that this was all serendipitous error, and that they were trying to put electrodes in other places in the brain. And when you do electrodes, you do these now with complex stereotactic maps. At that point, those didn't exist, but you basically use landmarks on the skull to tell you where you are, right? How are you going to find a target consistently? Well, you make measurements from fixed points on the skull, and these are always done. Remember that the skull is made from multiple independent bone plates during development that fuse, okay? And those fusion points give you two landmarks, bregma, which is the anterior central midline fusion point, and lambda. And that the story is that Olds misread the instructions and made his measurements relative to bregma instead of relative to lambda, and therefore was far more anterior than the electrode was intended to be, and it ended up in a region that gave them this phenomena. He observed something unusual and followed it up with the appropriate experiments. It now is clear that lots of structures give rise to this behavior, even though the medial septal region was the earliest. But if we think about it, and I'm going to wire them up a little bit more for you in the next bit, we know most of them, okay? So in addition, we have the nucleus accumbens. We have the ventral pallidum. I'm going to talk in a moment about the bed nucleus of the stria terminalis, okay? We have lateral hypothalamic bundles, okay? We have pedunculopontine tegmental region. We have the VTA, and we have lateral hypothalamus, and then a bunch of deeper nuclei further down. So all of these are in drive circuitry and behavior that leads to you feeling like you just got a reward. The rat sits there and taps the lever because it is doing nothing but feeling satisfied about itself for what has just happened, okay? Now, that's mostly happening to you, not in situations where you tap a lever, okay? You tap a lever and nothing happens most of the time. But what if you were getting the intense satisfaction that you get from the most rewarding experience you could have every time you learned one more neuroanatomic fact, okay? Okay? So that's the goal. is for you to try and operantly train yourself on learning neuroanatomy. Okay, so these are self-stimulating regions. And why this happens is in part a combination of the way VTA and accumbens are wired, and then the input from components of amygdala that are not so much about the fear and withdrawal and anxiety response, but in fact much more about reward. So when we normally think about amygdala, we think about its standard complex, it's got these nuclei, but there's a component of it that we see that projects out. So remember, the projection fibers from much of amygdala run through the stria terminalis. Turns out there are nuclei within the stria, so not everything that leaves is in fact going just to a target at the distal end, hypothalamus or thalamus. But some of them in fact are reaching this other structure, and that other structure, if you label it, projects a bit more widely, and it projects out into the nucleus accumbens. Okay? So when you do a labeling experiment and you label the amygdala, you miss those. Why do you miss them? Because your labeling die goes to the end point, and you go, oh, it just got lost in the stria terminalis. You don't see that there's the next neuron there that you have to label to project to discover that amygdaloid information gets out to these other targets. And these circuits are really important for addiction, in part because of the impact of opiate binding on what is otherwise a reward system that you use to enhance and learn behaviors. At the end of the course, there'll be a whole lecture on the dopaminergic inputs and the timing and the physiology of this. But when we think of it anatomically, we think that beyond the normal amygdala, we've got this zone that runs out further, this extra amygdala that runs where? Well, it comes up towards hypothalamus, and in here, this is putamen, caudate, globus pallidus. Remember, there was that little bit of ventral pallidum that was on the other side of the anterior commissure. And that's in fact the part which is most tightly connected. So when we think about accumbens, it's striatum. Okay? Has what are core and shell. They're based on staining in rodents. In humans, it's not clear that these really exist. But the accumbens is far more complex in its circuitry than caudate putamen. And that's why it's really a very separate beast. What else do we know that's separate about it? Big one? Where does it get its dopaminergic input? VTA, not nigra. Okay? So, it has inputs from the VTA, that's its dopaminergic. It doesn't get much input from motor cortex. It's not really about motor. Where does it get it? It gets it from the components that tell you about context and prior experience and how you feel about that, which are hippocampus and amygdala. Okay? So what you're really getting is not just, am I about to move my arm, but much more about what sort of things should I be doing? What do I know is coming? What do I know I just experienced? Did I just do this, my phone is ringing. Is this me sitting in my room surrounded by it and I should answer it? Is this me in a room with a patient and I will ignore it? Okay? You need to know that. You hear the same phone ring in both, okay? But you judge what to do with them differently. And you have to make a decision, and you have to gate that decision. And so your cortex is always being gated in a way by basal ganglia here. Anything that is decision-making or about output. And so you're doing that. And so the inputs come from those structures, and outputs go to hypothalamus and back to VTA, as well as up to some neocortical areas in prefrontal. So when we think about how these work, we've got inputs that come from basolateral amygdala, from hippocampus, and to some extent from the prefrontal cortex into accumbens. That's our striatum, okay? Its dopaminergic component comes from the VTA, okay? So now we've gotten inputs in and a dopaminergic system. But what are the steps and the rest of the loop? So what do we know a basal ganglia loop has to have? We start at the top. We have a input, which say for the putamen would be what? Primary visual cortex? Yeah, motor planning areas, right? Because this is helping you decide to make a movement, okay? And that goes to where? Putamen. So it goes to a striatal compartment. That goes to where? Well, it goes to pallidum, GP. It can go to GPE, and if it goes to GPE, where does it go? And then to and then to, and then, and that's the loop we built, right? And what are the little bits of variation that can come into play? What's the alternative for GPI? SN what? Right, the reticulata. Okay, and where does the dopaminergic projection go predominantly? Putamen. There's some to pallidum and others, but that's the major dopaminergic projection. So now we've got a different dopaminergic projection, different inputs. And in fact, when you think about this, the nucleus accumbens uses as its pallidal components the ventral pallidum, either going through the subthalamic nucleus or not, okay? And then it uses, to a large extent, reticulata instead of GPI. So it uses ventral pallidum instead of externa, and it uses reticulata instead of GPI. But it's exactly the same loop, and the thalamic major output is to mediodorsal, and where does mediodorsal go? Massive prefrontal cortex. So you've taken cortical input with all that hippocampal and all that amygdala that gives you the context for it, and now you've done the exact same basal ganglia manipulations and sent it back up to cortex, so cortex can use that, okay? So where is ventral pallidum? Well, I like this picture because you can actually really see that it looks just like pallidum. So if you look, here's pallidum, that's ventral pallidum, that tiny little slice there. Down here's where we'll find nucleus basalis and all, but this is ventral pallidum. If you took the anterior commissure out of the way, you wouldn't know that they were different structures. They are made up of comparable neurons, comparable density, comparable size, comparable packing, just different inputs. Okay. So if you look at how these interact, there's a negative interaction between anterior ventral prefrontal cortex and what's going on in the accumbens and the VTA. So prefrontal cortex is helping you decide to defer things. The VTA and the accumbens are telling you, great, reward right now. So the degree of negative interaction, the more they two are able to go in opposite directions, the better individuals, this is a functional MR study, the better they were able to predict success in waiting for a distant goal, okay, deferring immediate gratification, and they also turned out to scale with a measure of impulsivity on a trait scale, so that the better you are resisting an impulse, the more likely you are to be able to perform the task which requires you to wait, and therefore the more likely you are to have a really strong negative correlation between activity in the prefrontal cortex and activity in the VTA and accumbens. So the take-home message for you is to use your anterior prefrontal cortex for the next week, okay, not the VTA and accumbens, in order to actually study for the exam, okay? This is the best example I can think of, a distant goal as opposed to immediate gratification, okay? So I would tell you, though, on Thursday night, it is fine to have a party and enjoy yourselves. Do not try and cram on Thursday night. It's only going to make you miserable then and on Friday. Let me just say a couple of words about Wednesday and Friday. So Wednesday, we're going to do something, I've tried it a couple of times, and I think it works reasonably well. We'll spend about an hour in here going through a couple of clinical scenario cases. What I will post will be a set of slides where each slide has a little bit of a clinical history. When we do it in class, I'll put it up, we'll read it, and then I will pass out group numbers sort of in rough clusters to you, and I will randomly call on a number and ask you as a group to discuss where you think something is coming from. You know, the patient comes in and says they have a headache that affects this, and they turn out not to be able to move their left hand. Why? Where do you think it would be from? And then we'll go through that and work our way through that patient's clinical course. We'll do at least 2 of them. If I can organize a third and we have time, we'll do a third. After that, and the goal there is not to think about what the disease is, but to think about where things are where are lesions or abnormalities causing symptoms. Okay? You should have a good enough sense of organization of the whole nervous system now to be able to know the difference between what you would expect if somebody has a weak left hand because of a lesion in their spinal cord versus a lesion in the pons versus a lesion in the cerebral hemisphere. Each of those would expect it to travel with something different. So you're going to use that knowledge to say, well, what else is wrong? Where will it be? Do I find the other things that make sense? Okay? So that's the test. Don't worry about what the disease is. So take a look at them and think about them, but I'm really going to be asking people to partially think on the fly. There will then be a review session in lab. It's a very structured review that really tries to hit things that I think have been important over the course. Again, look at that in advance. Don't expect to come in, and please don't arrive with your list of 14 most pressing questions, because your pressing questions probably aren't anybody else's pressing questions. If you have them, email them to your TA or me or something, but really try and use that to use Wednesday's lab as a consolidating event. And then we will all gather on Friday. But I will see you Wednesday, and I'll post those probably tonight, assuming I get it written, and decide whether we can do a third one or not.